So our next talk is Dr. Xiaona Liu, who's coming to us from Yale University. She's going to be speaking to us about the novel large deletion mouse model of Angelman syndrome and how it can help us in drug development. She is the first FAST-funded fellow, and we are very excited that not only is she the first fellow, but she's actually done a tremendous amount of work, probably more work than many career neuroscientists have done in their entire lifetime. So you have done unbelievable work in the last 2 years, and we're so proud of you. So please come and share with us everything that you've learned. Forward, backward, laser. You could speak here or with the mic, whatever you prefer.
Yeah.
Want the mic?
Yeah, I want a mic. Yeah, thank you. Okay, shall we start it? Thanks for this opportunity. I want to talk about our new mouse model. Our new large deletion mouse model in the perspective of how we generate it and the difference of this model compared to the classic UB30A exon 2 deletion mouse model. I do not have disclosure. More than 70% of the Angelman patients are lack of the maternal contribution from chromosome 15q11 to q13, about 6 meg base. I know a lot of people already know this. However, the established UB30A exon 2 deletion mouse model and the UGD mice cannot represent this majority of our Angelman syndrome patients very well. The UB model only recapitulates about 5% of the Angelman patients, while the UGD mice is very good to study the GABA cluster, but there's no patients have the just UB30A, ATP10A, and the GABA3 deletion. Therefore, to fully understand the Angelman syndrome, we need a new mouse model which carries a 6 meg base large deletion on the maternal chromosome to recapitulate the majority of the Angelman patients. Here is a plan. We can use a CRISPR genome editing to insert the first FRT on the upstream of the large deletion region. After it goes to the place, we will use the same strategy to insert the second FRT. When both of them in the place, we can generate founder mice, which carries both FRT and the same chromosome. And then we cross these founder mice with an acting flap male, which can detect the 2 FRT and remove all the genes between the 2 FRT as long as the 2 FRT on the same chromosome. This means we will have a large deletion mice in the next generation. Here is how we perform this procedure. The entire process started on the female at 3 to 4 weeks of age. Before their natural hormone cycle is established, we inject the hormone PMSG to promote more eggs growth. 48 hours later, we inject the HCG, another hormone, to help those generated eggs to release. 2 hours later, we let this female to meet males. 16 hours later, this female should be pregnant, and each of them can carry 20 to 30 zygotes, which are the fertilized eggs. Then we collect zygotes from the pregnant females for genome editing. Insert these edited zygotes to the ovarian tube of a pseudo-pregnant CD1 mouse. We prepare those pseudo-pregnant CD1 mice through the same time window. We set them with those males which sexually active but no sperm generated. If everything goes as planned, 21 days later, we should be able to get the genome editing pups. And then I started to do the math, how many mice I need to get just one large deletion founder. We need 15 young females to generate 200 to 300 zygotes, and another 15 CD1 mice to be pregnant with these zygotes to get just one FRT in place. However, I need a founder carry 2 FRT, which means I need to get 15 1 FRT mice to repeat the entire procedure to insert the second FRT. If we do the math accurately, it will be 450 mice I needed for this procedure. Also, we were very lucky for this process. We use the mice less than the number by the end. Finally, I got the founder with a 2 FRT in place and excitingly set it with the acting flap male, expecting for the large deletion pups. However, instead of getting the large deletion pups, I got bunches of mosaic pups due to the large deletion-related complicated gene recombination. Until the 4th generation, I finally got a clean large deletion mice, and the deletion banner was sent for the single sequence. We confirmed the joint 2 sites of this 6 meg-based deletion. Then we did RT-qPCR using mouse brain tissue. We observed the expression level of the UBE3A, which is only maternal expressed in the neurons, is remarkably reduced because of paternal suppression. And the non-neuronal cells still have a little bit UBE3A expressed. The expression level of paternal expressed gene are not impacted by our large deletion. And the biallelic genes are expressed half in the large deletion mice compared to the wild type, which is expected. Then we conclude the large deletion mouse model is successfully generated. We also generate another 5 companion lines which had intact imprinting center and a UBE3A, also using the CRISPR genome editing strategy. The ASIC-A line is an intermediate product. It carries 2 LOXP sites. We cross these A mice with actin cream male to generate the B lines. The B lines has all the imprinting center downstream gene deleted. We use it to study the paternal expressed genes. Based on A lines, we insert another FRT upstream of UBE3A to generate the C line. C line is also an intermediate product. I cross these C lines with actin flap male and generate the D lines, which deletes upstream gene of UBE3A, including the entire GABA cluster. And this mice is a very good model to study the hypoinsufficiency we learned this morning, could use for the CRISPR-A method. So I cross the D lines with actin cream males and generate the E line, which is the final mice we needed. These lines, especially the E lines, are very important for further gene therapy study. They will be used as positive controls compared with the large deletion mice with or without gene therapy. And therefore, we have all the design lines generated, and we will look at the comparison of the large deletion mice with the classic UBE3A exon 2 deletion mice today, but all other lines have already started their behavior test. The large deletion mice are evaluated from the behavior test, electrophysiology, gene structure, transcriptome, and also molecular perspectives. According to the correspondence of the symptom of our Angelman patients and the phenotype of a known Angelman mouse model, we select the fear conditioning and novel object for cognitive function test, and the rotor and open field for the motor function. We also select the marble bearing and also the open field to assess the natural behavior function. We also did the fluorine-induced seizure, LTP, and EEG. A battery of the behavior tests were performed in order at a given time point. This is the same assay we use for the gene therapy study. In the open field treated on the 3 to 4 weeks of age, both large deletion mice and the UBE mutant mice have reduced total travel distance in the 1-hour test, and the reduction of the 2 Angelman mice are quite similar. However, when we retest at 8 weeks, this shows the mutant mice reduce more total travel distance compared to the large deletion mice. However, this is somewhat unexpected. We do not think this is implied the large deletion mice perform better than the UBE3 mutant mice for the locomotion. From the working with these mice for quite a long time, I actually hypothesized, or a more possible explanation is that the large deletion mice has a different type of behavior. It's like the arousal is unique to this line. As shown in the video, already finished the video. There's a little mice. It's rampaging. It's almost flat if you haven't run into a little mice. This observation is confirmed by the rotor test 2 weeks later. Also, both AS mice have significant reduced latency on the rotor in both accelerating and steady paradigm compared to the wild type. The large deletion mice stand on the rotor for a little bit longer time than the deletion mice. And the marble bearing test reflecting the natural behavior function shows the same deficiency of large deletion mice and mutant mice. The novel object recognition is one of the cognitive functions. The mice were exposed to 2 familiar objects for 5 minutes. 30 minutes later, the mice will be introduced to the same arena again with the familiar objects on one side and a novel object on another side. The time they spend with the object counted. 24 hours later, the mice will be introduced to the same arena again with the same familiar object on one side and another novel object on another side. Again, the test revealed the similar cognitive deficiency of these 2 lines, but long-term memory of the large deletion mice may be a little bit more impaired compared to the mutant mice as shown in the right panel. Both large deletion mice and UBE3A mutant mice are susceptible to seizure induction. The seizure sensitivity of the large deletion mice is more compared to the UBE3A mutant mice, showing the shortened latency for the myoclonic seizure and tonic seizure. This is consistent with the observation we found in the Angelman patients. I should mention that the spontaneous seizure was observed in large deletion mice, which we have never observed in the mutant mice since we work on these lines for many years so far. But we still plan to do the EEG study to see if continued EEG recording could be a better way to assess the seizure quantitatively. We know the UBE3A mutant mice has impaired synapse plasticity by electrophysiological recording of brain slice from mice. The recording was used to analyze the long-term potentiation of hippocampus A1 to CA3 region using high-frequency stimuli. Similar to UBE3 mutant mice, the LTP is impaired in large deletion mice, although we do not use the UBE mutant mice for comparison here. In summary, we have successfully generated the 6 MAC-based large deletion Angelman syndrome mouse model confirmed by the molecular and behavior test. The comparison between the new large deletion mice and the UBE3A point mutation mice indicated a hyperactivity, hypersensitivity, and increased seizure sensibility of the Angelman syndrome for the first time. We also evaluate additional behavior impaired unique to the large deletion mice and need more study on them. We also generate another 5 companion lines can use for further study. Finally, I want to thank my mentor, Dr. Yonghui Jiang, and thank FAST Foundation provides me fellowship.